We are continuing today to look at hemoglobin structure and function. Previously, we had finished with nucleic acids. I think we did nucleic acids, the structure. Then we even looked at function, going into protein synthesis, gene regulation. We finished all of that. So today we are continuing to the next topic, which is on hemoglobin. So we'll look at the structure and function of hemoglobin today. The next time when we meet, then we'll continue to look at hemoglobinopathies, diseases that are related to abnormalities in the structure. A bit of historical background. Blood has been known to carry oxygen, say at least since about, say, 1793. So that's like in the 18th century. Blood has been known by, older scientists who have been working in the field, have had the idea to know that blood carries oxygen. So that can be seen from people like, communications of persons like William Withering. William Withering was a British physician, also a chemist, physiologist, even though he was well known for some work he did in finding the active ingredient in a herbal product, which, because he himself was a medical doctor and found out that one of his patients used some herbal product, which went into studies to know the active ingredient which was in that mixture. And so he is well known for like the work he did in the finding of the active ingredient in that mixture. It became clear that there was an active ingredient in a plant, digitalis. Today, it is out of his work that you have a drug called digoxin, which is used for the treatment of myocardial infarction. And in some archives, some of his letters that he was writing to some of his colleagues, he made mention of, in those times, what is referred to as factitious airs. So factitious air simply means artificial air. So not natural, like, just like you have now, say you have oxygen tanks that you use in the clinics, for instance. But in the first time, factitious air was referred to as, referring to hydrogen gas, which was, separated. So he referred to this, factitious air in blood, like some interactions. His notice of, say, factitious oxygen air is mentioned somewhere here. That in applying such, administering such gas, there was a certain effect. So he documented some of these things down. So right from that time, newer scientists referred to some of these archives and began to work on it, knowing that blood was carrying oxygen. Right. Then there was another British, also a physician, also a doctor, who also, though a doctor, at the same time he was a prolific writer. So he wrote a lot of articles, and in some of those articles you could see him also referring to factitious airs. He worked very much on treatment of tuberculosis, pneumonic conditions. And so, of course, that is to do with the respiratory system, respiratory tract. So he also made mention of some of these, factitious errors in the treatment of some favorite conditions, things like that. Then fast forward to about 1851, you have a German molecular biologist, as a biochemist, that was able for the first time, to crystallize hemoglobin. I mean, that's a way of, let's say it's extraction of blood proteins. And how did he do that? He did that with solvents like water, alcohol, petroleum ether. Was able to extract, the protein just by mixing blood with these solvents, and then with some slow evaporation of these solvents, he was able to have a crystal form of the blood protein, hemoglobin. So he reported that also in his publications. Various publications in 1851 were on his works of crystallization of hemoglobin. But still, the structure of hemoglobin wasn't known until you had Max Perutz. He was British, all right, but he was born in Austria. So you see that he was working in the University of Vienna in Austria. And at the same time, he was also working in a university in England as well. And at this time, there was already a new method had come. There was the emergence of X-ray crystallography, which could help to determine the structure of molecules. So he was able to determine the structure of hemoglobin, and together with John Kendrew, also another British, I think he should be a Scottish or so. He was a crystallographer at the same time, a biochemist, and he worked on the structure of myoglobin. We'll come to see myoglobin in addition to hemoglobin. So you see the similarities. So that in 1962, the two of them, shared a Nobel Prize in chemistry, which was for their work because it's hemoglobin and myoglobin, it was termed as the finding of the structure of heme-containing proteins. Because we come to see in hemoglobin, we have the components referred to as the heme. So that is, a brief historical background of hemoglobin. I just feel sometimes some of these things, when you just start to study them, you should appreciate the fact that it started from somewhere. People worked, did several things. And so when you just see it now, and it's being applied in various fields and it's working. That some people struggled in the past, working on these things for years before later others also worked on it and things became clearer and clearer to us now, the younger generation who are studying these things. Right, before we continue, I want you to understand that you can have proteins categorized into two major classes as globular proteins to hemoglobin, if you listen to the name, it says hemoglobin. So you should understand that it's a protein, and that protein is globular. It's a globular protein. That's why it's called globin. The beginning, hemo, the heme. So the heme, which is a prosthetic part, we'll come to see. So together, heme, globin coming together to give you the name hemoglobin. Because so hemoglobin is not a fibrous protein. A fibrous protein is fibrillated proteins. With let's say fibrinogen, for instance, or fibrin, which forms a mesh over, say, a broken skin, a wound, in order to stop blood clotting. That's a fibrous protein. Or maybe some muscle, proteins like, the tendons. Those are fibrous proteins, but you have hemoglobin being a globular protein. All right, so that is clear why the name is such as a globin. Now we can continue to look at some basic overview of hemoglobin. You've had some historical background, and hemoglobin is that pigment in blood responsible for the red color of blood, and is found in the red blood cells, what we refer to as erythrocytes. The amount of, say, hemoglobin you'll find in males and females really doesn't have a significant difference. If you look at it, for females, about 13 to 15 grams per deciliter; for males, 14 to 16 grams per deciliter. You may say there is some slight difference, to say that in males it's a little bit higher, but really, if you look at it, these numbers overlap, and so there is really not a significant difference in amount of hemoglobin in males and females. It's almost the same. And you have known hemoglobin to be responsible for the transport of oxygen from the lungs to the tissues, and then the transport of carbon dioxide from the tissues back to the lungs, and then it is exhaled out of the body through the nostrils. These basic things you have known already. So today we are just coming to build on some of this knowledge which you already have. Now we can move on to take a look at the structure of the hemoglobin molecule. Now the hemoglobin molecule is a tetrameric protein. What it means is that hemoglobin is made up of 4 protein subunits. What you see being depicted here as alpha 1, alpha 2 subunits, beta 1, beta 2 subunits. So you have hemoglobin, being known to have these 4 subunits. But of course, the structure which you have here is actually just the most common. So the commonest of the hemoglobin structures you'll find in adults, which is the hemoglobin A1 structure. Because you also come to see that we actually have different types of hemoglobin, but this is the commonest. This is what you find mostly in adults. And so when the structure of hemoglobin is being referred to, this is what is mostly used to depict the structure of hemoglobin. Now you also know that you have proteins and some non-protein molecules that are in complex together. Like if you talk about enzymes, for instance, you have a protein, which is an enzyme, and you can have a metal, like say copper, magnesium. And these act as what we refer to as cofactors to these proteins. And we refer to the protein as a prosthetic part of that molecule. The same way for hemoglobin, you have the protein. So what you see is the blue lines and then the black lines refer to the protein chains. So that will be, the peptides. And then the red boxes inside them are referring to the prosthetic part, which is the heme. So that heme is not a protein, but it is in complex with these protein subunits. The molecule will be non-functional. And so is tetrameric and also allosteric at the same time. Then you have also known the structure of proteins to be made up of amino acids. So you remember in first year biological chemistry, you talked about the structure of proteins, where you have the primary structure of proteins, which is the sequence of amino acids in the peptide that you are referring to. So it means that you can actually step many amino acids make up the structure of these peptides. And you see in the alpha chains, the alpha subunits, you have 141 amino acids that form the alpha subunits. And you have two of them. The beta chains also contain 146 amino acids. So when you put these numbers together, you realize that in the structure of hemoglobin, you have 574 amino acids. So that is if you are studying the primary structure of the protein, the globin. You realize you are having 574 amino acids that make up the hemoglobin structure. Now, because you have studied proteins already, you have known the structure of proteins, primary structure, secondary structure, tertiary structure, quaternary structure. We'll not be going into that so much about the protein structure of the hemoglobin. What will be focused on is the structure of the heme, because you already have the structure of proteins. You have already studied that, and so if you know that it's made up of 574 amino acids, together to give you the structure of the proteins. So we'll move on to take a look at the structure of the heme. The structure of the heme. That is what you are going to add up to what you already know about the structure of proteins. Now, first of all, you have what we refer to as other ring with a nitrogen, at the tip there. That's a pyrrole. It looks like, if you study carbohydrates also, you saw something like a pyranosis, for instance. This is, so you have a pyran. A pyran is having an oxygen in place of nitrogen there. That's a pyran. And this, what you are seeing here, is what is referred to as a pyrrole. It's having a nitrogen there. Now, what you see here is that you can actually have a synthesis of a pyrrole just by amination of pyran, and then you have the pyrrole. But what we'll be going to now will be that you'll see that these pyrrole rings actually can come together to form what we refer to as metalloporphyrins. So first we'll see, say, the porphyrin to together with some metals is what you find in many pigments. So it's not even only just in hemoglobin. Maybe in the next lecture, when we can, we will see that you have even some other pigments, even in plants, for instance, that will have similar structure. Can take a look at the pyrrole ring. I see when I change the slide, I am monitoring it on my phone device. I see that whilst I'm speaking, it takes some time before the slide changes. So that's why sometimes I wait a little bit. All right, so you have the structure of the heme, what I was referring to earlier, that you have 4 pyrrole rings, which are labeled A, B, C, and D. So if you look at A, just take a look at that nitrogen, and you realize that nitrogen is in a ring that is a pyrrole ring. When you take a look at B, you also see that B, the nitrogen is also a pyrrole. That's a third pyrrole. You see the nitrogen is in a ring. And D, the fourth ring, is also a pyrrole. You see the nitrogen there is also in a ring. So that's a pyrrole. So you have 4 pyrroles that are connected by what we refer to as methynyl bridges. So you see these methynyl bridges that are, binding these pyrroles together to form a ring structure. And you see in the middle of the ring structure is the ferrous ion, Fe2 plus ion. You have known that blood contains iron. So the ion this ferrous ion, is having interaction with these nitrogens that are inside these pyrroles. And actually, it is that ion, the ferrous ion, on which oxygen is carried. So that is shown there, that oxygen is carried on that ion. And at the same time, that ion also have interaction with the side chain of the amino acid histidine, what you refer to as an imidazole ring. So you see the side chain being so you have a particular histidine molecule which actually is bound onto the ion. So that's what is being depicted there. Then also you have some letters that are written on each of the pyrrole rings. Each of the pyrroles is having some letters M, V. Mean M means a methyl group, so CH3. V is a vinyl group. And then the P is a propionyl group. So you have these carbon chains that are attached on the pyrroles. Looking at the structure of the heme in detail, you'll see this. And the pyrrole, the full structure, what you are seeing, the ring is referred to as a porphyrin ring. And what you are looking at is actually what is being referred to as a protoporphyrin. So you have this porphyrin ring, which is made up of 4 pyrrole rings as the structure, the ring structure of heme that is having the metal ion, the ferrous ion at its center. Right. Now, I mentioned earlier that we have different types of hemoglobin, but the structure that we have been talking about is what you find, mostly in adults. That's the HbA1 hemoglobin, which is composed of 2 alpha and 2 beta protein subunits. And you see here, the total hemoglobin percentage, which you find in adults, is approximately 90%, almost 90%. So around 90%, you have that hemoglobin A1. That is why a lot of to be saying that it has 2 alpha chains, 2 beta chains. But you have also hemoglobin A2, which is having 2 alpha proteins, 2 delta proteins. We'll come to see what this means, what beta, delta, we'll see. And there you can see that the amount that you find in blood, in adult blood, is less than 5%. Then we have the fetal hemoglobin. So in the fetus has a different type of hemoglobin, which is different from us, the adults. Remember that the fetus in the uterus does not breathe oxygen from air, does not breathe in air to take oxygen through the lungs. It takes blood from the mother through the placenta. And so the placenta has a barrier, and it is the fetal hemoglobin which carries oxygen, which is from the blood of the mother to the fetus. And that fetal hemoglobin is different from the adult hemoglobin. It's made up of 2 alpha chains and 2 gamma chains. But of course, at birth, after we are born, over a period of maybe 24 to 48, 72 hours, our hemoglobin gets converted after there is that exposure to air, to oxygen. You have the change of conformation, change of the fetal hemoglobin to the adult hemoglobin, to the 2 alpha, 2 beta chains. Of course, there are some rare disease conditions where some people still have some large amount of fetal artificial oxygen that the person will be put on, or even some blood transfusions to deal with such problems. But these are some rare disease conditions. There too, you see in an adult, like I was saying, the fetal hemoglobin gets converted. But of course, you can have in us still some amount of fetal hemoglobin, less than 2%. And now you cannot, we also refer to, it's an adult hemoglobin, all right, but it is what we refer to as glycation. It is conjugated with glucose. So you have some glycated hemoglobin, also referred to, which is less than 5%. Because glucose has some complex forming with hemoglobin, that is glucose in blood. And those of you who work in some clinical settings have known that you have some glycated hemoglobin tests for diabetic conditions. That is, this test which is taken over a period of some months, maybe 3 months. An average is looked at to confirm a diabetic condition or not. It is based on this glycated hemoglobin. Right, so we have these different types of hemoglobin. Then also, apart from I realized my network went off for some few seconds. So I stopped talking for some time, waiting for it to come. Right. So I was saying that, in the history, we were referring to when the structure of hemoglobin was determined with X-ray crystallography, and the Nobel Prize that was won by Perutz and Kendrew. Kendrew was working on myoglobin, and together hemoglobin, myoglobin, they are heme-containing proteins. Now, you can take myoglobin as, say, if you are taking just one subunit of a hemoglobin. Of course, it's a different molecule altogether on its own, and that is made up of 153 amino acids. It's monomeric. It's only one subunit. So one subunit, protein with heme in it. We'll come to see also its function. As a reservoir and some intermediate between hemoglobin and the tissues. So it's not like hemoglobin, which is carrying oxygen from the lungs to the tissues, but myoglobin is in the tissues. And when hemoglobin brings the oxygen, it does it, uh, oxygen, particularly in damaged cells that require oxygen rapidly in the process of healing or repairing of these cells. You find myoglobin acting there actively. Okay, now since we are talking about uh, hemoglobin and myoglobin being responsible for the transport of oxygen, we can talk about the binding affinity of hemoglobin to oxygen, the same way myoglobin, oxygen. Now, if we take a look at these two curves, the red curve is for myoglobin, the blue curve is for hemoglobin. Now, these curves are showing relationship between saturation of the myoglobin and hemoglobin molecules in relation to concentration of oxygen. But here because oxygen is a gas, we refer to it as a partial pressure of oxygen in millimeter mercury. Now, what you notice is that, you see, myoglobin is a monomer. So if it's a monomer, it means that it will get what? I mean, there are some notes here. Look at it. One hemoglobin molecule can carry 4 oxygen molecules. 4. Because it's a tetramer. And we saw from when we're looking at the structure of the heme, we saw that the ferrous ion at the center was carrying 1 molecule of oxygen. And you have in each of the protein subunits 1 heme molecule, which means each of them can carry 1 oxygen molecule. So together, you have the hemoglobin molecule carrying 4 molecules of oxygen. But myoglobin is a monomer, is a monomeric protein. It's a monomeric structure and can carry only 1 molecule of oxygen. So what it means is even at low concentration of oxygen, the oxygen, you see, at very low partial pressure of oxygen, myoglobin already gets saturated. It already gets saturated up there. But you see, hemoglobin, that there is rather a gradual rise of saturation, gradually, all the way, before it gets in the lungs there being shown, the partial pressure is high of oxygen because you breathe in air from the nostrils, which contains oxygen. Oxygen is rich in that air. So there's that high partial pressure of oxygen. To understand, you say high concentration of oxygen in the lungs. It is there that you find hemoglobin getting saturated. And in the tissues where hemoglobin will be releasing the oxygen molecules, you see that concentration of oxygen there is lower. So you have partial pressure of oxygen in tissues and then partial pressure of oxygen in the lungs. So you can relate these of having hemoglobin being tetrameric because it can carry 4 molecules of oxygen at a time. Imagine that it was monomeric. You see that you will have less efficiency in doing its work of carrying oxygen to the cells. You may need to have a lot of these hemoglobin molecules to be able to carry out its function. But with four subunits, it's able to carry 4 molecules of oxygen, which is much more than that's 4 times higher than what myoglobin does in the tissues. So that's a comparison of binding of oxygen and oxygen to hemoglobin and myoglobin, the two of them, in relation to partial pressure of oxygen, or say concentration of oxygen. Now we can talk about the mechanism of binding of oxygen to hemoglobin. You see, we were just talking about the fact that hemoglobin is tetrameric. It's made up of 4 subunits and is carrying on the heme. Each heme molecule is carrying 1 molecule of oxygen, and together will be carrying 4 molecules. So how does this happen? It's not like all the 4 molecules of oxygen come and attach to the molecule at once. No. We have what we refer to as cooperative binding of oxygen to hemoglobin. In other words, the binding of oxygen to hemoglobin is cooperative. What does it mean when we say it's cooperative? So we're looking at from left to right, the binding of oxygen to hemoglobin in the lungs, from left to right, because that's what is showing the binding of oxygen to hemoglobin. Now, you see, first, you have just 4 boxes there. That is to depict the molecule, the tetrameric molecule of hemoglobin. That tetrameric molecule of hemoglobin in the lungs, first, 1 molecule of oxygen comes and binds. Now, the moment that oxygen binds to the subunit, it increases the affinity of hemoglobin for more oxygen. And so a second molecule comes to bind to a second subunit, which increases the affinity of hemoglobin the subunit. That also will result in increase more of the affinity of hemoglobin for oxygen. And then the last molecule. This is where you have the hemoglobin molecule being saturated with oxygen. All its subunits are bound to oxygen. is oxygen. One binds, increases affinity. Another one increases affinity the more. Now, when you are looking at this mechanism in the opposite direction, that is from right to left, that process will be occurring in the tissues. That is where oxygen carried by hemoglobin has to be released to the tissues. And so you see, when you have one molecule of oxygen released, so if we are looking at it from right to left, one molecule is released. When one molecule is released, it reduces the affinity of hemoglobin for oxygen. And so a second molecule of oxygen is released to the tissues. Then a third molecule is released to the tissues and is left with the last oxygen molecule. Affinity is reducing, and so oxygen is released again, the last. Before you have an oxyhemoglobin, that's a hemoglobin saturated with oxygen. And hemoglobin that now releases the oxygen to the tissues become deoxyhemoglobin. So that's what we are referring to as the cooperative binding of oxygen to hemoglobin. All right. Now, we are talking about affinity of oxygen to hemoglobin being increased or reduced. This brings to what we refer to as the T and R forms of hemoglobin. T only means taut or tense, or just say tight together. And R means relaxed. So when you see T, it means tense, tight together. R means relaxed. Now, two beta subunits. Now remember, when you were of proteins in first year, tertiary structure of proteins, what were the bonds that you found in the tertiary structure? You refer to hydrogen bonds, you refer to disulfide bridges, you refer to salt bridges, you also refer to hydrophobic interactions. So these were some of the I think I've mentioned all of them, the bonds which you find in the tertiary structure. And it is just these tertiary structures that come together as subunits to give you a quaternary structure, like the structure of the hemoglobin that we are talking about. Now, if you pay attention here, you see that between the alpha and the beta subunits, you have some strong hydrophobic bonds. Strong hydrophobic bonds. So that's a bond that you find in give you the quaternary structure that you are having. And you are also seeing that you can have some weak hydrogen bonds. The alpha subunits. some weak weak hydrogen bonds are found between the alpha subunit. So these bonds are responsible for, bringing that whole quaternary structure together. You have some strong hydrophobic bonds and you have some weak ionic and hydrogen bonds bringing the structure together in a tense form, in a taut form, in a tight form of hemoglobin, where there is, so as I mentioned, a deoxyhemoglobin, no oxygen at all. Now, what you see is that the binding of oxygen. So if you pay attention, you realize that at the center here, the heme is being shown, with the ion, which is violet. And you see here too, we have a depiction, the blue that is binding onto the ion, that is oxygen. And you see that oxygen, when bound to the hemoglobin molecule, disrupts these weak hydrogen or ionic bonds. So we see oxygen disrupts this bond, which makes the hemoglobin molecule relaxed. So that explains why when one molecule of oxygen binds to a subunit, the affinity of the hemoglobin molecule increases. And so a second molecule comes to bind. Bonds, making the molecule relaxed to have a higher affinity for oxygen. Right. Now we can talk about, the mechanism by which the transport of oxygen and carbon dioxide by hemoglobin, and even to some extent myoglobin is involved, how this is carried out. We see here the lungs, the circle at the top depicting the lungs. In the lungs, that is where you have oxygen coming in, breathed in. So on the right-hand side, oxygen, breathed in. Containing red blood cells. Red blood cells have hemoglobin. The hemoglobin is having heme. And in the heme, you have central ferrous ion. On them, you have oxygen molecules. 4 oxygen molecules bind to the hemoglobin molecule. So that is an oxyhemoglobin. That oxyhemoglobin moves with the blood to the tissues. Now the circle below here depicting the tissues. Now in the and look at this, there is myoglobin there. See, when oxygen is released from hemoglobin, myoglobin picks that oxygen molecule and releases it to the cell. So that's why I was saying myoglobin plays the role of a mediator. It picks the oxygen molecule one after the other from hemoglobin and transfers it to the cells. And I mean, what's the major cellular respiration process that you can refer to? Let's say the process of generating energy from glucose, for instance, the process of glycolysis, the Krebs cycle, things like that. And you have processes, carbon dioxide. And the carbon dioxide byproduct is picked by the hemoglobin molecule and moving with the blood back to the lungs, carbon dioxide in the lungs, then exhaled out of the nostrils, out of the body as a byproduct. That is produced in cellular respiration. That is carried by the hemoglobin molecule back to the lungs to be exhaled out of the body. Only 15%. So what happens to the 85% of carbon dioxide, which is produced as a byproduct? What happens to it? All right, so let's move on to look at what happens to the 85% of the carbon dioxide that is produced. Now, on the left-hand side is what is happening in the tissues. And on the right-hand side is what happens in the lungs. As far as what happens to the carbon dioxide is concerned, that's what we are talking about. That is the 85% of the carbon dioxide which is produced as a byproduct. Now, that 85% of carbon dioxide, most of it will react with water. I said most of it because, of course, if you are looking at biochemical processes, as a whole, you actually come to see that carbon dioxide can also have some other functions apart from what we are just looking at. Because you can have some carboxylation reactions and all of that. So some of these can go into those reactions. But you have most of these 85% of carbon dioxide, which is being produced, reacting with water. The enzyme that is acting in that process is carbonic anhydrase. And the product which is formed is carbonic acid. So you have carbon dioxide, water reacting to produce carbonic acid. Carbonic acid then dissociates into bicarbonate ion and hydrogen ion. And that should quickly tell you, hydrogen ions being produced means that pH will be reducing, more acidic condition will be created. That's what it means. But quickly, hemoglobin, which is bringing in oxygen to the tissues, releases the oxygen molecules and picks the hydrogen ions. So it means that hemoglobin molecules pick some of the carbon dioxide as a molecule carbon dioxide and transport them to the lungs to be exhaled. Some of the hemoglobin molecules bind to hydrogen ions. And that hydrogen ion, together with the that is bound to the hemoglobin molecule, is carried to the lungs. How about the bicarbonate ions? The bicarbonate ions move unbound freely in blood, so that upon reaching the lungs, hydrogen ions are released by the hemoglobin molecule whilst it picks oxygen molecules from the lungs to take to the tissues. And the hydrogen ions that the hemoglobin molecule has released then binds to the bicarbonate ions. So this is a reverse of the reaction that was occurring in the tissues. So that you now have hydrogen ions and bicarbonate ions now reacting to give you carbonic acid, gives you back the carbonic acid, and that carbonic acid then breaks down to carbon dioxide and water, get exhaled. So that's what we say, that carbon dioxide is exhaled with water vapor out of the body. So that's what happens to the 85% of carbon dioxide that is produced as a byproduct of cellular respiration. You have just 15% carried directly by the hemoglobin molecule, and you have the 85% going through this process of formation of carbonic acid, which then breaks down to bicarbonate ions, hydrogen ions. The hydrogen ions are moved, bound to hemoglobin molecules, whilst the bicarbonate ions move unbound in blood. Upon reaching the lungs, the hydrogen ions are released by the hemoglobin molecule, which would then react with the free-moving bicarbonate ions, forming carbonic acid back. And from it, you have carbon dioxide and water formed back and exhaled out of the body through the nostrils. Now, with that said, we have what we refer to as allosteric effectors. So these are uh, substances that facilitate the release of oxygen to the tissues. So the oxygen that hemoglobin is carrying from the lungs to the tissues. Already we refer to what the concentration of carbon dioxide or concentration of oxygen as partial pressure. So partial pressure of carbon dioxide is one of the things that facilitates the release of oxygen. Of course, in the tissues, that is where you have cellular respiration producing carbon dioxide, which means carbon dioxide will be in higher concentration in the tissues. And the concentration is what we are referring to as the partial pressure of carbon dioxide. And that high concentration of carbon dioxide facilitates the release of oxygen. So because hemoglobin quickly wants to release the oxygen so it can pick the carbon dioxide. We also saw just now the process of the 85% of carbon dioxide which is produced, which will have to react with water molecules to form carbonic acid, which then breaks down to form hydrogen ions and bicarbonate ions. And I quickly mentioned that it means you are having a decrease in pH. You are having an acidic condition being created. That's what it means. Now, that acidic condition also facilitates the release of oxygen from the hemoglobin molecule. If that is not done, of course, it means that the blood will become very acidic, and that can be detrimental. So, in this condition of high pH due to hydrogen ions, oxygen quickly is released by the hemoglobin molecules and quickly binds to the hydrogen ions. Now, remember that all these processes that we are referring to, say cellular respiration, metabolic processes that are generating energy. These processes are occurring inside the cell. So the cells that make up the tissues that the blood has flown to, the processes are occurring inside of the cell. What it means is that carbon dioxide we are referring to and the hydrogen ions we are referring to, they are coming from outside, they are coming from inside of the cell to the extracellular space. That's what it means. Because you have cellular respiration occurring, mostly, let's say, it's occurring in which organelle? In the mitochondria. That's where you have energy generated. And if you are having hydrogen ions being released from the cell to come to the extracellular space, that's an ion carrying positive charge. What it means is that if that charge, which is coming out of the cell, is not compensated for, it will create a potential across the cell membrane. It's like you are having one side becoming more positive than the other. So it will create a gradient across the cell membrane, which means it can cause some of the hydrogen ions to be going back. Like hydrogen ions will just be in a circle going back, be forcing to go back because there's that potential. One side is positive, one side is negative, and so there will be that movement. And so because of that, you have chloride ions which are carrying negative charges compensating for that. So that potential is not created across the cell membrane. And so chloride ions also moving out of the cell to the extracellular space will be compensating for that. And so it's also referred to as an allosteric effector. It is also facilitating the release of oxygen from the hemoglobin molecule. Now, you have what you refer to as 2,3-biphosphoglycerate. I think you did some metabolism of carbohydrate in first year biological chemistry. You saw the process of glycolysis. If you go back to that process of glycolysis, you see that one of the intermediates in that process is this 2,3-biphosphoglycerate. I think first you have 1,3 or 1,6 biphosphoglycerate, and from it you have this 2,3-biphosphoglycerate. Now, why is this an allosteric effector? I think we will come to that, why this 2,3-biphosphoglycerate is an allosteric effector. Basically it's because it binds to the hemoglobin molecule and actually disrupts those weak ionic bonds, those weak hydrogen bonds we were referring to between the alpha globins. In a way, it means that it changes the conformation of the hemoglobin molecule to the R form, which means that it reduces affinity of the hemoglobin molecule for oxygen. So, in other words, oxygen will be released to the tissues. We'll talk more about 2,3-biphosphoglycerate. We'll talk more about it. Now, with pH, what we just described, that you have, reduction of pH facilitating, the release of oxygen, is represented by what we refer to as the Bohr effect. And we are just looking at it again with this oxygen dissociative curve that is showing the saturation of oxygen, being influenced by the partial pressure of oxygen. The blue curve is the curve at a pH 7.2, which is lower, and the red curve pH 7.6. That's some slight difference, which means the blue curve is for a more acidic condition than the red curve. Now, what do you notice? You notice that you require a higher amount of oxygen in the case of the pH 7.2, the lower pH, that is the more acidic condition, in order for the hemoglobin molecule to become saturated. It only means that you are having a lesser affinity of the hemoglobin molecule for oxygen. And that actually is what facilitates, the release of the oxygen molecule, so that it can now, pick carbon dioxide and take it back to the lungs to be exhaled out of the body. Now, as I told you, I promised we will come back to the 2,3-biphosphoglycerate. Actually, I also mentioned that the 2,3-biphosphoglycerate is produced in the process of glycolysis. And the process of glycolysis is a process that occurs in the cell all the time, generating energy. So the 2,3-biphosphoglycerate binding to hemoglobin, it's something which is in normal blood. You have 2,3-biphosphoglycerate binding to hemoglobin in normal blood, at a certain normal level. It's there to regulate the affinity of hemoglobin for oxygen. And here again, when we're looking at the oxygen dissociative curve, you realize, so the blue is referring to a normal blood with 2,3-biphosphoglycerate. The red curve is referred to blood that is without the 2,3-biphosphoglycerate. And you realize for that blood that is having no 2,3-biphosphoglycerate, affinity for oxygen is higher. That's why it gets saturated at a lower concentration of oxygen than for the normal blood. But you see, in anemic conditions, the cell has some adaptive. It's an adaptive response to anemic condition, or in some hypoxic conditions of where there isn't enough oxygen, much oxygen. The cell will produce more of the 2,3-biphosphoglycerate, which bind to the hemoglobin molecule. It is to make the hemoglobin molecule more relaxed, so that its affinity for oxygen is reduced, so that when it carries the oxygen from the lungs, it will release it faster to the tissue cells. Now, you'll find this, usually when you hear things like, when athletes are preparing to go for a tournament, sometimes the Ghana Black Stars, for instance, maybe they go to Kenya, say they want to go to a higher altitude, where there is a less atmospheric pressure, so that they have some adaptation increased, because when you come from that higher altitude to a lower altitude where you have a higher atmospheric pressure with high amount of oxygen, the body begins to perform the more. It is because there is slightly a hypoxic condition that is created at such high altitudes. There is lesser amount of oxygen at such higher altitudes. And the body in that process of adaptation, which I refer to, produces more of these 2,3-biphosphogylates, which bind to the hemoglobin molecules, disrupting the weak ionic and hydrogen bonds between the protein subunits. And you see that the biphosphoglycerate is having 2 phosphate groups carrying negative charges, and you see positive charges that are on the subunits, the protein subunits of the hemoglobin molecule. So there is that, kind of, it goes there and sort of disrupts the bonds, which usually the bond is between the alpha chains, these what you refer to as the weak ionic and the hydrogen bonds, so that you have rather the R form of hemoglobin. And so you have, the affinity of hemoglobin for oxygen reduced. And so you have that affinity rather for carbon dioxide. Carbon dioxide will be picked from the tissues. Oxygen will be released faster. So that is some scientific, background of such an application, even in athletics, in sports. So you see, it's practical. It's something which is not out of reach. It's practical. It's studied, it's known, and is used even to improve, performance. Now, I think we can end with some derivatives of hemoglobin. So we'll look at 2 derivatives of hemoglobin which can occur. So for instance, you can have what you refer to as methemoglobin. And methemoglobin simply refers to hemoglobin in which you are having oxidation of the ferrous ion to a ferric ion. That is Fe2 plus converted to Fe3 plus. Fe3 plus will not carry oxygen. And so if the ferrous ion is oxidized to the ferric ion, the hemoglobin molecule loses its functionality. And that can be caused by some free radicals, some oxidants like hydrogen peroxide, and even some drugs. So some drugs like I think these lidocaine, for instance, some drugs which are used even in the treatment of even some antibiotics like sulfonamides, for instance, some antimalarials like chloroquine, for instance, they can cause some of these, oxidation of the ferrous ion in hemoglobin, which then will cause some, loss of functionality of hemoglobin. But the good thing is that the body quickly, reacts to this. It has a system by which you have an enzyme, methemoglobin reductase, which quickly, reduces the ferric ion back to the ferrous ion. So the body has that mechanism in place that even if there is that oxidation, it will quickly. For you to have methemoglobinemia, it means that there must be very drastic condition that will have such a case where you have Fe3 plus in hemoglobin in very large amounts, at high amounts. What that will result to is that it means that the hemoglobin will not be able to carry enough oxygen to the tissues. Then what it means is that it will result into some form of a feeling of breathlessness, suffocation. What you feel maybe along the chest, things like that. Maybe even some vomiting, head turning. Maybe some people may even have some convulsions, or may fall unconscious. Those are serious conditions that can result into that. Now, the second, and I think this should be the last slide, is what we refer to as carboxyhemoglobin. Carbon monoxide. You have air pollution coming from, engines all over the place. Carbon monoxide is actually having like 200 times more affinity to hemoglobin than oxygen, which means when carbon monoxide binds to hemoglobin, it's really difficult for that carbon monoxide to be released by hemoglobin. So, clinically, persons who are acutely exposed to carbon monoxide, what is done is to put them on oxygen, because it's like some sort of competition of carbon monoxide and oxygen molecules for hemoglobin, so that when you increase the concentration of oxygen, it goes to displace the carbon monoxide molecules. And the carbon monoxide is released from the hemoglobin molecules, and over time, the person will get well. But of course, that's, again, as I mentioned earlier, you can cause headache, nausea, vomiting, breathlessness. It's a form of suffocation. Enough oxygen is not reaching the cells. And so there's a combination of these symptoms that can be felt by such persons. So if you know some of these things, it's good. You make sure you stay away from lots of pollution all over the place. Our university is close to Agbogbloshie. I think they are still burning a lot of things over there. And as a scientist, should be concerned about some of these things, how people are burning rubbish all over the place indiscriminately. Farmers just get up and start burning the bush indiscriminately because you have some weak systems to control some of these things. All right, so I've been talking. Now I'll keep quiet and listen to you. The floor is open. If you have any questions, so far no one has written in the chat. If you have any questions, you either write in the chat or you can put up your hand. I will enable your microphone, call your name, you speak, and I'll respond. So the floor is open for you.